Hello everyone. Welcome back here to the OS Student Research Grant Seminar Series. We are about halfway through here, I think, as we start uh, our seminar today. So I want to welcome everyone. My name is Robert Driver. I'm a postdoc at Duke University. And as I say each week, each year the AOS offers funding to students and early career researchers with the AOS Student Research Grant. And here at the AOS Student Research Grant Seminar Series, we celebrate the challenging and innovative work of the grant recipients by sharing AOS-funded or related work to the AOS members here. So each week we're showcasing the research plans of a 2024, 2023, or 2022 AOS Student Research Grant winner in this seminar-style format. And we have one seminar each week for 15 weeks. Like I said, we're at the middle here. And I want to thank everyone for being here today. Thank the AOS and AOS Student Affairs Committee, Chris Ann Handel and Ramona Fletcher for their support and input, and especially thank Haley, our speaker today. So before we turn our attention to Haley's research, I wanted to mention if you have any questions, there will be time for a Q&A session at the end of Haley's talk. And you can participate by unmuting yourself. Oh, every week I say that, sorry. You can participate by raising your hand with the hand raising icon, and I will unmute you if you would like to speak. I'm sorry. Or by typing anytime during the hour in the chat box or in the Q&A box, and I will monitor all three of these question asking ways to make sure we get all of our questions in. I'd like to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. I will post a link to that code in the chat. And also, I will post a link to the post-seminar evaluation form where you can provide feedback to us about the seminar, and that just helps us improve the seminar week to week. And I'll also include a contact email for the Student Affairs Committee if you would like to reach out to us. And please be aware that we are recording this talk. This week, Haley G is currently a master's student in ecology and evolutionary biology at University of Arizona. And Haley's academic career began with a double major in Bachelor's of Science in Biology and Biochemistry at Oberlin College in Ohio. And following graduation, Haley worked in a variety of ornithology and wildlife field technician jobs across the United States and Illinois, as well as working with condors in California and with puffins on the very remote island of Middleton Island in the Gulf of Alaska. In 2023, Haley began graduate school in the lab of Renee Duckworth at University of Arizona, studying the effects of maternal stress on offspring development and behavior in Western Bluebirds, which we will hear about today. And so with that, I'll turn it over to Haley for her talk entitled Endocrine Mechanisms Behind Unadaptive Maternal Effect, Linking Breeding Site Quality to Offspring Behavior.
Okay, thank you so much.
Yep.
So to start, each new generation is impacted by resource allocation from the prior generation. So this is true from single-celled organisms with splitting of the cytosol and molecules and materials in the cytosol up until larger multicellular organisms that provision offspring through things like eggs, seeds, or internal development of offspring. This resource allocation fundamentally has an influence over an individual's traits because these resources are really the first building blocks that make up an individual's development. And these effects may be even more pronounced in organisms that have a longer or prolonged period of development where offspring are reliant on resources from parents. And these resources from parents, this parental allocation is in turn shaped by ecological context. And so here, ecological context can be things like food availability or competition that not only impact sort of the limits of what the prior generation can provide to the next, but also the internal physiology of the parent in different contexts. Costs of stress response and maintenance of homeostasis will be different, affecting internal physiology of the parent and then what they pass on to offspring. So putting these two things together, you have this potential for information to be transmitted from one generation to the next. And the maternal environment, through those maternal resources, can influence offspring traits. And here I'm going to start saying maternal because I'm focusing on maternal effects today. So thus, there's an opportunity here for offspring traits to be impacted by an environment that they haven't even experienced yet of their own life. And one question I'm asking here today is if these resources can function as cues for adaptive maternal effects. We know that there are these really important building blocks for development, but do they contain information above and beyond that, allowing for sort of specific adjustment of traits to environmental pressures? So the function of these resources is going to be really important in thinking about how maternal effects can actually evolve. But considering these three sort of steps along this pathway, each of them is complex, right? Each step is multi-component, so thinking about how the environment can affect maternal resource allocation, things like food availability, stressors, and competition will all have different effects there. Maternal resources is this very complicated landscape, so it includes stuff like nutrients and energy for starting development, as well as signaling molecules like hormones. So here I have glucocorticoids and androgens. And then offspring phenotype, there's a lot of traits that could be influenced by maternal resources. Things like body size, physiology, and behavior are sort of all on the table there. Um, so here, there's a lot of information to parse between each of these steps. I like each of these arrows here. And one question I'm asking, thinking about this large amount of information available and how maternal effects evolve to sort of use this information is there integration or regulation of information along this pathway? Our maternal resources and offspring traits, or sort of the allocation of resources and development of traits, paying attention to all of the information they can from the previous step, or is only certain information being used in the evolution of these maternal effects? And if there is this regulation of information, at which step along the pathway does it occur with these arrows here? So another way to think about this is sort of what are mom and offspring paying attention to uh, in the evolution of these maternal effects. So thinking about how maternal resources reflect the environment, are diverse environmental inputs distilled in maternal resource allocation, or is mom sort of filtering what information about the environment actually affects her resources that can affect offspring? And then thinking about how offspring traits respond to maternal resources. Do these resources have links to individual traits, or do they jointly affect an integrated phenotype? So what aspects of maternal resources do offspring traits remain developmentally sensitive to? And different answers to these questions are going to have very different influences on how maternal effects can actually evolve. Um, so to understand this broader pattern of adaptive maternal effects, we have to know how these links evolve in between these sort of steps in the pathway. So now I'm going to explain to you, I have three different hypotheses sort of talking about the characteristics of these links and how they might relate to broader maternal effect evolution. My hypothesis one is this master regulator hypothesis, which has this idea that offspring traits are jointly influenced by a master regulator resource that responds to the maternal environment. So here I have some sort of more generalizable predictions. Here I have nutrients shown as the master regulator resource. And here I'm thinking about that resource as something that's sort of developmentally very important for the organization of multiple offspring traits. So the prediction here would be as the quality of the environment increases, you have more allocation of nutrients to offspring, which that high allocation of nutrients to offspring will have effects on body size, behavior, and metabolism. Larger body size, changes in behavior, and lower metabolic efficiency. And so if we see maternal effects evolving in this pattern, we see offspring traits covarying, they're constrained to covary due to constraints between the relationship of the resources and the offspring traits. My hypothesis two is this covariation hypothesis. So here it says that maternal resources covary in response to their environment and jointly influence many offspring traits. The predictions here would be as environmental quality increases, you have sort of a coordinated change of multiple different maternal resources. So for example, under a high quality environment, you might have more nutrients, lower glucocorticoids, and lower androgens provided to the offspring. And then as you have sort of that axis of coordinated changes in maternal resources, those have effects on multiple aspects of offspring traits. So here, when you have sort of all of the things you get when the environment is good versus the things you get when the environment is bad, when the environment is good, you get a higher body size in offspring, predict changes in behavior, and lower metabolic efficiency. And if we see maternal effects evolving in this way, you still have that sort of constraint or covariance of offspring traits, but it's due to this constraint on maternal resource variation rather than, so the earlier link in this pathway. And then my last hypothesis is the specificity hypothesis, where specific aspects of the environment can influence individual maternal resources that are linked to a single offspring trait. So here in these colors, I have examples of like 3 potential different specific pathways. And then on the bottom, I have this prediction with the androgens. So as you have higher competition in the environment, you have higher allocation of androgens to offspring. And then with those higher allocations of offspring, you predict higher aggression, of androgens to offspring, you predict higher aggression in offspring. But yeah, you could see sort of these predictions for any different traits. I mean, if you have maternal effects evolving in this way, resources may function more as cues, allowing for offspring traits to be adjusted by specific aspects of the environment. Um, so we might expect something like this in like a predictive adaptive maternal effect where the environmental pressures offspring will face can be predicted by the environment, the maternal environments. And I kind of see this hypothesis allowing for more independence of offspring traits and maternal resources. And then overall, these hypotheses sort of on a scale of like more constrained to less constrained, with 1 and 2 being more constrained, but at different points in the pathway, and then 3 being a little less constrained. And then sort of as a preview of the results, we're actually going to see a combination of different hypotheses with aspects of specificity and aspects of covariation. All right. So, how am I going to actually test these hypotheses? And I have to sort of be able to measure things about the environment, maternal resources, and offspring traits. And luckily, my lab works in this Western Bluebird system, which is a really great system to look at these things because it has this well-established maternal effect. So you expect connections between those links there. And then it's also pretty amenable to that sort of multiple sampling that's necessary. So overall, this maternal effect established by my lab links maternal territory quality to offspring behavior. So I'll sort of break that down for you and then tell you more about the individual parts that I'm measuring today. So aspects of territory quality, which are determined by sort of nest box occupancy in these birds, influence maternal stress. And when territory quality is poor, the breeding females have been shown to have higher circulating baseline glucocorticoids, so they're stressed. And then those stressed females lay eggs with higher amounts of androgens in the eggs. And then those stressed females also produce offspring that have these behavioral differences with increased aggression and dispersal coming from females on poor quality territories. So we know these sort of different puzzle pieces. We know that androgens are important. We know that behavior is important. But thinking about my hypotheses and sort of this multi-component nature of these different parts along this pathway, I'm really curious to see how other aspects or components of the egg are involved in this maternal effect. And then looking at the effect on offspring, if this effect is more of sort of a specific adjustment of behavior, or if it's an adjustment of a wider offspring phenotype where behavior is sort of one of the traits involved here. And thinking about other traits that might be involved in a maternal effect, one thing that might be related to maternal stress and maternal resource allocation is functioning of the HPA axis. So the HPA axis is the hypothalamic pituitary adrenal axis. It's basically this sort of communication system between 3 different organs. You have stimuli being processed by the hypothalamus, which through a couple of sort of hormone cascades stimulates the adrenal glands to release glucocorticoids into the bloodstream. And one of the main functions of glucocorticoids is to regulate sort of metabolism and maintenance of homeostasis, energy balance in the body by increasing the amount of glucose available in the bloodstream. But during times of stress, glucocorticoids can also serve to sort of organize different systems of the body to prioritize returning to allowing the animal to sort of attempt to return to homeostasis. So for example, they can decrease energy going into reproduction and increase energy going into self-maintenance pathways during times of stress. Um, and to measure glucocortic or HPA programming, we can look at both HPA reactivity through stress-induced glucocorticoid levels after an animal has been exposed to a stressor, or we can look at baseline glucocorticoid levels to sort of look at that normal function of glucocorticoids in a non-stressed animal. Another trait that might be important thinking about maternal resources is body size, which could be influenced by both maternal resource allocation and sort of early life behavior in these birds. So now that I've introduced my study system and some predictions, or some offspring traits, I'm going to give you some predictions and then sort of talk about my methods and results. Um, So here I have these pathway predictions on, there'll be sort of three, one for each of the hypotheses. So on the left, I have aspects of ecological context. So I have territory size and competition. In the middle, there's variables representing maternal resource allocation, so nutrients in the egg, glucocorticoids and androgens. And then on the right, I have offspring traits listed. So I have HPA reactivity, body size, and baseline glucocorticoids. And these pathways, these arrows between the different variables are predictions of the relationships that will exist under these different hypotheses. So under this master regulator hypothesis, you have this sort of very important, developmentally important resource. And here I have egg nutrients predicted as that resource, which vary with ecological context and then influence sort of a coordinated effect on many different offspring traits. HPA reactivity, body size, and glucocorticoids. Under this covariation hypothesis, I have ecological context influencing the allocation of multiple maternal resources at once, sort of this coordinated allocation of resources, which then has an effect on many aspects of offspring traits. And then under the specificity hypothesis, you have more specific links between environment, maternal resources, and traits. So here I have competition influencing yolk androgen allocation, which then influences HPA reactivity. But you could also see predictions sort of for other aspects of offspring traits under this specificity idea. And I wanted to also sort of return to this constraint idea that we have the specificity hypothesis, which is sort of the least constrained, and then the top two are more constrained, but at different points along the pathway. So the first part of what I'm going to test is this link between the environment and maternal resources. So I'm going to give you some predictions here, directional predictions. Under this master regulator hypothesis, like I said, nutrients are sort of predicted to be this master regulator resource. And then as territory quality increases, you have higher allocation of nutrients to offspring. Under this covariation hypothesis, as territory quality increases, you have a coordinated change of multiple maternal resources. So higher amounts of nutrients, lower amounts of glucocorticoids, and lower amounts of yolk androgens at a high territory quality. And then under this specificity hypothesis, as specific aspects of the environment change, you have an influence on a single maternal resource. So here, as competition increases, you have higher allocation of yolk androgens would be the prediction. So I'm going to talk to you a little bit about how I measured the ecological environment variables and maternal resources, and then talk about my results for that relationship there. So going back to this sort of overall idea of this maternal effect, we know it starts with territory quality. And in these birds, bluebirds are cavity nesters, and the presence or the availability of cavities is their main limiting resource for breeding. and they will breed in these nest boxes that we provide for them. And previous work has shown that manipulation of nest cavities is an effective way to induce this adaptive maternal effect. And this is because bluebirds have these fierce interspecific competitors for cavities. In Montana, where my fieldwork takes place, it's tree swallows. Um, and the tree swallows will come and harass the bluebirds at their boxes, and the bluebirds need to defend their boxes, which is stressful. But if you set up, or if there is another nest box close by to your focal bluebird pair's box, a tree swallow pair will move in there, and basically they will sort of defend the whole area, including our bluebird box area. And they will sort of buffer the focal bluebird pair from further competition from the tree swallows. So previous work in this lab has shown that bluebird pairs on double box territories face lower intrusion rates than bluebird pairs on single box territories. So it's this really cool way to sort of manipulate competition for our birds. And in my project that I'm talking about today. This was the summer of 2024. I did all my fieldwork for this project, and it's at two sites in Western Montana. 26, I had 26 breeding pairs total, and I made 14 of those pairs into double box territories by setting up an extra box once the birds started to nest. And then I left 12 as single box territories. And in addition to this competition with having the extra boxes, I also looked at territory size, because territory size may reflect availability for the breeding pair. Bluebirds are insectivores, and they'll kind of use whatever area is open in their territory to forage. So having a larger territory may allow for better access to food resources to feed themselves and to feed their nestlings during the breeding season. Um, so those are the sort of two aspects of the environment I measured for this project. And then thinking about maternal resources, I'm really focused on what's going on inside of the eggs. So I collected from each of those boxes the third laid egg before incubation started. And one resource we're looking at is nutrients, which may be reflected in egg size. And there is sort of a pretty good variation in egg size with these eggs. Here I have an example. This is like normally they don't really look like this, but this is an extra big egg compared to a more normal sized egg for you to be able to see variation in egg size. So to measure egg size, we measured the mass of the whole egg and also the mass of the yolk and put those into an egg size principal component with lower values, meaning smaller eggs and smaller yolks, and then larger values, meaning larger eggs and larger yolks. Another important resource I'm thinking about is steroid hormones. So female birds deposit steroid hormones into their egg yolks, and yolks are formed in the left ovary of the breeding female bird. This is a histology section of a chicken ovary, and you can see these circles are the yolks that are being formed. And the yolks will accumulate hormones from the cells directly surrounding them, where sex hormones will be provided through those cells, and then we will also see uh, accumulation of glucocorticoids from the adrenals to the bloodstream, and then those are incorporated in the ovaries. So with our egg yolks, I dissected out the yolks from those eggs, which is this picture here with this little round circle on the left, and then halved those egg yolks to look half for hormones and half to look at yolk formation differences. So to look at hormones, I used liquid chromatography mass spectrometry to identify and measure hormones in the egg yolks. And all in all, we found 13 different hormones. These are the names, but not super important that you remember all of them. Because we saw that these hormones tended to covary with their functional classes, so I grouped them by class and covariation in principal component analyses. And what we got out was that we have a progestogen, and estrogen PC, which contained all of the hormones from that respective class. We have a larger androgen PC, which has 5 androgens. And then we have this lone androgen in the bottom here, this orange one, transandrosterone, which is an androgen but didn't covary with the rest of the androgen PC, so that one is sort of left separate. And that one will actually be pretty important for my results coming in a minute. And then we also have these two glucocorticoid metabolites, TH-CORT and deoxyCORT, which also didn't covary with any other hormones. So those ones were also left separate for analyses. The other thing I did with that other egg yolk half was me and this awesome undergrad in my lab, Katie Cannon, stained the yolks to be able to look at yolk formation and the dynamics of yolk formation. So egg yolks are formed by the female bird. She adds sort of a light layer and a dark layer of lipids every 24 hours. And staining allows you to both count, the number of layers and also look at the thickness of those layers. Um, so with those two measurements, we created a yolk deposition speed PC with lower values, meaning slower yolk formation. So more time, more days of yolk formation and thinner layers, and then larger values, meaning faster yolk deposition. So fewer days and thicker layers in the yolks. Um, all right, so I looked at hormones, nutrients, or egg size, and then yolk ring staining for my sort of maternal resources. Um, and I'm going to return to my predictions before telling you about my resources. Results. So as a reminder, under this master regulator hypothesis, as you have better territory quality, you predict higher amounts of nutrients or that master regulator resource. Under this covariation hypothesis, at higher territory quality, you have sort of a coordinated change of many different maternal resources. And under the specificity hypothesis, in response to specific environmental pressures, you have change in individual maternal resources. So here, as competition increases, you have higher yolk androgens. And what we found looking at the relationship between the environment and these maternal resources was that actually only one of our maternal resources had a direct relationship to the environment. And it was this transandrosterone, that sort of lone androgen that didn't fit in with the rest of the androgens in the PC. And it related to territory size. And what we noticed was that a lot of the birds just put 0 transandrosterone in their eggs, and then we had a group that put sort of a higher amount. So I dichotomized this variable and found that there's a significant difference in territory size between the 0 or low transandrosterone eggs and the high transandrosterone eggs, territory size for the female bird. So transandrosterone was lower on larger territories and higher on smaller territories. And we did look at the other maternal resources and did not find any significant relationship to the ecological variables. But we did see a trend with the androgen PC, that's like the rest of the androgens that weren't our transandrosterone androgen, were higher on territories with higher competition. And this result fits in with what we know about our maternal effect. Previous work has shown that androgens, sort of as an overall class, were involved in the maternal effect, that females from poor quality territories laid eggs with higher androgens. But with this method, we're actually able to have a little bit higher resolution and see that maybe it's not just all androgens total, maybe it's this one androgen metabolite, transandrosterone, which I think is really interesting. So I looked a little bit into the function of this androgen, transandrosterone, or just androsterone. And overall, it's sort of considered to be maybe a weaker androgen. But in mammals, it seems to have this specific function during development of masculinization of fetuses. So maybe this androgen has this specific function during development beyond sort of its general function as an androgen in like adult physiology. In addition to looking at the links between environment and maternal resources, another thing I was thinking about with those hypotheses is sort of the relationship between maternal resources to each other and the constraints or covariation that might be going on there and how that will influence offspring traits, maternal effects on offspring traits. So when we look at that, the relationships, how maternal resources co-vary with each other, we see that yolk deposition was correlated with other resource measurements. So we see in these figures, I have yolk deposition speed on the y-axis with slower values on the bottom, faster values on the top. And then on the left here, I see that those higher transandrosterone level eggs had faster yolk deposition. And then on the right in this figure, I see that smaller eggs also had faster yolk deposition. So here we see that yolk deposition is associated with multiple other components of maternal resources. And I looked a little bit more into this, and previous work from Alex Badyev in my department has shown that in-house finch eggs, when yolk formation duration takes longer, they have higher levels of vitamin E and carotenoids in their eggs. So I'm thinking that potentially yolk deposition may have this role in coordinating the covariation of other resources, sort of the mechanism for covariation. So putting all of that together, we see these links between the environment and maternal resources, that eggs from small territories have higher transandrosterone, and that higher transandrosterone is associated with faster yolk deposition, and that faster yolk deposition is associated with smaller egg size. So here we have elements of specificity, this link between territory size and transandrosterone, but then that is associated with potentially this mechanism for covariation in yolk deposition. So there's sort of this combination here. But in addition to individual eggs and what's going on with those, each egg itself, we know that female birds lay a whole clutch of eggs. So we looked at clutch size as potentially an important covariate, in that it may reflect overall investment in reproduction, and then it will also have an effect on sort of the post-hatch nestling environment by influencing how many siblings each nestling has and food competition and all of that. So in our birds, they lay between 7 and 4 eggs, with the modal number being 6. And looking at the results with clutch size, we see that clutch size increases with both measures of territory quality. So here on the left, on both of these figures, clutch size is the y-axis. On the left, I have territory size as the x-axis, and you can see that as territory size is larger, the birds tend to have larger clutch sizes. And then also looking at this figure on the right, you can see that clutch size is larger in birds that have that extra box on their territory, so that sort of buffer against competition on their territory. So putting that together, here we see that clutch size may reflect overall investment in reproduction by the female. All right, but the next step of this sort of figuring out these pathways is to look at how maternal resources might affect offspring traits. So yeah, what are the links between those resources and the offspring traits? And to measure offspring traits, I sort of returned back to those same boxes in my experiment and measured the nestlings. So each of the nestlings that are born in one of the boxes at our field sites, they get these unique color bands so we can identify them from afar. They get, a federal bird band, and they get morphological measurements. So the measurement I'm using today to look at body size is wing length. So that's what you'll see as a proxy for body size in the nestlings. And then I also measured aspects of HPA programming. So with the nestlings, when I got them, I would put them in a paper bag to collect a fecal sample, which is to represent our baseline or sort of integrated baseline glucocorticoid levels. And then after holding them in the bag for a little while as a stressor, I took a blood sample to look at stress-induced glucocorticoid levels or HPA axis reactivity. And I'll sort of use those. I think I'm mostly saying HPA axis reactivity, but I mean sort of the same thing. So some predictions about that link between maternal resources and offspring traits. Under this master regulator hypothesis, you expect with change of that important resource, here I have nutrients, you predict a sort of a coordinated change in many offspring traits. So here at higher nutrient level of allocation to offspring, you predict higher body size, lower HPA reactivity, and lower baseline glucocorticoids. And the overall effect there would be phenotypic integration or covariation of those offspring traits. Under this hypothesis two, this covariation hypothesis, remember we have this sort of axis of coordinated change in maternal resources. And sort of along that axis, you expect a change of many different aspects of offspring traits as well. Um, so as maternal resources covary, like when territory quality is high, you'll have a combination of maternal resources that predict higher body size, lower HPA reactivity, and lower baseline glucocorticoids. And here we also see phenotypic integration on the offspring side, but it's due to sort of a constraint on the variance of the maternal resources themselves. And then with this specificity hypothesis, you expect specific links between maternal resources and offspring traits. So here I have, as yolk androgens increase, you have higher HPA reactivity. And this hypothesis allows for more independence of traits in the evolution of these maternal effects. All right. So the results here, first looking at body size, we saw that body size responded to yolk deposition speed, yolk androgens, and egg size. So with this figure here on the y-axis, I have wing length, and then there'll be like two other more figures that are going to pop up with the same y-axis, but with different x-axes. So on the x-axis here, I have yolk deposition speed, and we see that as uh, there's faster yolk deposition. The nestlings have smaller wing length or smaller body size. This result here, we see that as egg size increases, you have a larger body size with the nestlings. And then this last result here, we see that as androgens increase in the egg, you have larger body size. And this is not that transandrosterone, that this is the larger androgen PC. And overall, we also looked at the relationships between other maternal resources and body size and did not find relationships between glucocorticoids or transandrosterone and body size. Um, so we see this multi-component resource transfer impacting body size with these relationships. And then we also looked at HPA programming and found that these baseline glucocorticoid levels increased with faster yolk deposition. So here you can see that relationship with yolk deposition speed on the x-axis and baseline glucocorticoids on the y-axis. And we did, so here that sort of implies that clutches with faster yolk deposition produce nestlings with the capacity for higher metabolism or potentially higher levels of glucose in the blood, higher glucose metabolism. And I did look at the other maternal resources in relationship to baseline glucocorticoids and did not find any relationships there. And then we also looked at HPA reactivity and didn't find any really significant relationships between HPA axis reactivity and the maternal resources, but we did see a couple trends. So one trend was that HPA axis reactivity decreases with larger clutch sizes, and that HPA axis reactivity is higher for female nestlings. And overall, we did not see correlation between offspring traits that we measured. Um, All right, so putting those results together with the maternal resources and offspring traits, we see that nestling body size was influenced by multiple resources, and that we also have a link between yolk deposition speed and baseline glucocorticoid levels. So putting these in the context of my initial hypotheses, um, these results support a combination of the specificity and covariation hypotheses without support for a master regulator, the master regulator hypothesis. So we have this specific relationship in orange between territory size and yolk transandrosterone, where at smaller territory sizes, we have higher amounts of yolk of transandrosterone in the egg yolks. And that may hint at some sort of buffering by the mother, the female bird, of sort of what information about the environment is relevant to influence maternal resources. Um, and we also have this aspect of covariation. So yolk transandrosterone was associated with yolk deposition, which may have this role in coordinating other resources. And we see that some of those coordinated resources influence offspring body size along with androgens. And then we also see that yolk deposition influences baseline glucocorticoid levels. So overall, we do see that some of these traits of body size and HPA programming may be involved in this adaptive maternal effect. But we didn't actually study aggression, which is sort of the focal trait of this maternal effect. So that leaves this potential for this specific pathway to still maybe influence behavior or aggression. And I'm wondering if that specific pathway may involve transandrosterone, because we know that androgens are important in this maternal effect. And we did have this effect with territory size and transandrosterone, but transandrosterone was not directly linked to any of the offspring traits that we did study. Um, so looking into that potential for relationship between aggression and our maternal resources leads me pretty nicely into my next steps, which are looking at embryo brain gene expression. So in addition to those eggs I collected to look at maternal resources, I also collected some late stage embryos to look at gene expression and use the brains from those embryos to do RNA sequencing. And I'm working through that data right now. But I'm looking for potentially the specific link between transandrosterone and aggression by targeting brain neuroendocrine pathways that are known to be involved in aggression and behavior and also HPA axis programming. So I'll be looking for differential gene expression in the serotonin, dopamine, and glucocorticoid pathways, in addition to sort of looking overall at the gene expression happening there. In addition to just looking at what genes are expressed, I also sectioned like a fraction of each of the brains. So we'll be looking at localization of those important genes that we identify by using in situ hybridization on tissue sections. So I'm really excited to get this sort of maybe uh, more fine-grained understanding of how behavior may be underlain by these changes in brain gene expression. All right, so that's my project that I came to talk about today, and I wanted to do some acknowledgements to my lab group, to my PI, Renee Duckworth, to Katie Cannon, the undergrad who helped with a lot of stuff, including the specifically about the yolk staining, and then Matt Jenkins, Juliana Costers, Alex Badiev, and Carmen Sanchez Moreno for a lot of helpful feedback about this presentation and project in general. And then a special thank you to my 2024 field crew, Molly Vander Werp and Cora McKinney. They were both awesome, and I know some of them might be looking for jobs, so if you hear from them, they're both great. And then I also wanted to thank my funding sources, especially AOS. The student research grant at AOS funded a lot of the hormone stuff, so that was super important. All right, so that's it for my talk.
All right, great. Thank you so much, Haley. Really exciting results and very exciting upcoming work to in your upcoming chapters as well. Um, so there are people clapping through emojis, but if anyone wants to ask a question in the variety of ways, I will monitor all those. And uh, I'll start out with something that I just think I might have missed. But how was the territory size measured?
Oh, it was maybe I forgot to explain this, but it's the distance to the next bluebird box. So the
Okay.
next box occupied by another bluebird.
Okay, and that's somewhat dependent on how you place them, or
That's more of the experimental manipulation is in the competition, like adding those extra nest boxes.
Mhm.
I mean, territory size could be influenced by like sort of generally how they're placed in the site, but I didn't
Mhm.
manipulate that in my study.
Okay. Um, yeah, I was just curious. And then I wanted to ask, so it's like
Mhm.
one thing leading to another in these pathways. But are there correlations between like the first and last domino? Like for example, I have this like long thing, but maybe it's not correct. Like the small territory, wait, no, the yolk deposition led to slow accumulation, which led to a large. Basically, I took the first domino and the last domino and found that small territory is associated with larger egg. Is that something that I can do by skipping across the pathway that you presented, or that doesn't work?
Um, I think we saw actually maybe just the directionality got a little confused, but I think with that link between um, territory size, yolk deposition, and egg size, you would expect the smaller territories to result in faster yolk deposition, which is associated with smaller eggs. Um, but yeah, I mean, we didn't see a direct relationship between yolk deposition and territory size. So I think it's like seeing maybe there's these links there, but yeah, I'm not totally sure about that. But
Okay.
yes, I did also look at sort of relationships between the traits and the territory size measurements. Um, and we do see some relationships there, but they're maybe not explained super well by the resources we measure. So maybe there are other resources or other aspects of the maternal effect that we don't explicitly measure here that are also important. There's also this sort of sex effect that I don't get into here because I can't measure the sexes in those fresh eggs that I collected. But that is potentially important in the maternal effect, the differences between the sexes.
Okay, gotcha. Um, and then I wanted to ask, so if you didn't cut the trans uh andrest I can't
Yeah,
say it.
uh-huh.
So
Androsterone, uh-huh.
yeah, you made it into a binary between uh in one of the analyses. It may have been
Yeah.
territory size.
Mm-hmm.
So if you don't turn that into a binary, is the result still significant just based on a correlation?
Yeah, it is still significant,
Oh,
but
okay.
it like performs overall better as a binary. So,
Okay, got you.
mhm.
Um, I have a very simple question. Was it the
Yeah.
third egg that you selected because that's just like the middle of the average clutch?
Yeah, previous work in the lab has shown that sort of there's these, there are differences in hormone allocation along the clutch size, but the middle one is sort of a good, yeah, showing of the average. And yeah, the average is 6, so we did the third one as the middle one. And then I didn't get into the embryo results because I don't have them yet, but collected the other one that was closest to the middle for the embryo to try to sort of get the best overall view.
Got you. And then the fledgling measurements were, or not fledgling, but the nestling measurements were then the other individuals in the nest.
Yeah, all the remaining ones.
Okay,
Yeah.
got you. The ones that made it through your guys, made it out of the egg. And then, so did you control for the effect of like multiple fledglings in a single nest?
Yeah, all of so I showed sort of raw data to have something to show, but all of
Mhm.
the analyses were mixed models. So it's like controlling for nested effects of like nests in site and all of that. And some of them take into account, depending on what was, best for the model, like nestling rank and things like that
Ah,
too.
got you. Got you.
Hmm.
That's great. Um, and then did you ever, maybe you don't have enough power, but did you ever look at like between the two territories, the two sites, if there were any differences?
Um, I've looked a little bit, not in these analyses, no. Site for like with the mixed models is taken into account like as a random effect, but I haven't looked specifically at what the the statistics between the sites. But yeah, our sample size isn't super big, so
Right.
mhm.
Yeah, I was thinking, and there's no reason for you to necessarily think any two sites in this part of Montana would be different for any reason.
No, these ones are pretty similar. I would wonder if there's differences between these sites and some of the other sites we work with. But these two sites are more similar. They're sort of both along like rural roads and sort of big in comparison to we have some sites that are more on like smaller sort of public parky areas.
Okay,
So I would wonder if there's differences between those, but I would think these ones would be more similar.
got you, got you. Okay, we have a question. Thank you for your talk. I may have missed this, but how were you quantifying HPA axis reactivity?
Oops.
Uh,
Um,
and then there's
right.
a couple more questions after that, which I can read now. Was it just stress-induced concentration? And if so, do you think that metric is meaningful without a baseline plasma concentration to compare it to understand the degree of change?
Yeah, I think that's a good question. Yes, it was just looking at stress-induced concentrations for the nestlings. We did take into account the baseline from the fecal samples, which I understand is a little bit different, but tried to take into account that way. Um, and we did look at those two measurements were like not correlated with each other, so we treated them as more separate traits. Um, but yeah, I think just due to sort of limitations on what you can collect from our little birds, that's how we ended up doing it.
And what was the amount of time that they were handled for
It was
in
about
that?
like 20 or 30 minutes.
Okay.
And yeah, they're at the age where you would expect, because nestlings kind of have a time where they have hyporeactive HPA axis responses, and they're old enough, they're like 15 days old, that you would expect them to have, and they did have that HPA axis response.
Cool. All right. So I'm looking across here. I think that's all the questions. I'm very excited for those RNA-seq and in situ hybridization analyses. So please write another AOS student research grant so we can have you back at the seminar series to report on those. But if those are all the questions, we're right about at 53, 54 after the hour. So we'll say that was a fantastic seminar from Haley, and thank you so much for your presentation, and thank you so much everybody for being here.
Thank you.
We will see you guys next week.